Hi. So today I'm going to talk about the central dogma of molecular biology. So in this, there are two processes that occur, transcription and translation. Let's first talk about what DNA is. DNA is deoxyribonucleic acid. This is genetic material that can be found in all living things. It acts as an information holder for cells and can only be found in the nucleus. So first, let's talk about transcription. This is when the DNA gene sequence is transcribed or copied to create RNA. RNA is ribonucleic acid. This provides the information to create a protein, which our bodies need. So in transcription, there are three steps. The first step is initiation. So this is when the DNA begins to separate. RNA polymerase attaches to where the transcription will begin. So this is going to be our DNA strand, and then I'm going to do the RNA polymerase right here. Sorry, I don't have a whiteboard. And then, so it's going to clump onto the DNA strands that are starting to untwine and separate. And then, so that's step one. So for steps two is called elongation. This is when the RNA polymerase slides across in that direction slides across the DNA strands and begins copying or creating mRNA. So like that. And it's copying this first strand right here. The third step is termination. This is when the RNA polymerase hits the end location. So there's a sequence right here that ends, for like a sequence of code that ends the copying and stops creating mRNA. This then lets the mRNA strand free, and this leaves the nucleus. All this is happening in the nucleus. So then once this is done coding or copying, this will be set free and leave the nucleus. And this will go towards the cytoplasm. So after that, the next we're going to talk about is translation. Translation is when the amino acids and proteins attach to the mRNA. So let me get rid of this. Hopefully you could see that. So let me draw the mRNA strand again. And then we'll have amino acids all around. So these here, amino acid. All right, so there are the amino acids in the cytoplasm, and then, so let's say the mRNA strand, one of the codes was like AGG. We're gonna have this occur in the ribosome. The ribosome is gonna go around it, and then we'll have some tRNAs out here. So transfer RNAs. So in this translation process, the tRNAs are bringing amino acids into the ribosome. So let's say this amino acid is attached to this tRNA. This tRNA is going to come into the ribosome, and look for a set of code on the mRNA that it pairs with. So if this one pairs with AGG, then it will connect right there, and the amino acid will be like deposited right there. So then let's say the next one is ACA. This tRNA, if it matches with ACA, this amino acid, it's attached to the tRNA, the transfer tRNA, and goes into the ribosome again, and then deposits its amino acid right here. And then this one gets pushed out. Once these tRNAs drop off the amino acid, it'll go back out and look for another amino acid that'll attach to it, and then continue that process, making these this protein chain of proteins. Hopefully you can see that. So transcription and translation are important because it allows our DNA to be copied, and it creates proteins for our body. So without these proteins, without this process, we wouldn't be able to copy our DNA or create proteins for our body. Thanks.